So welcome to SI. This is the Biochemistry Review. So if anyone who's not here for Biochem, it's all right if you stay in the room, but I'm just going to ask if you can keep the noise volume down to a minimum so the other students who are in the review can hear. That would be really awesome. So thanks in advance for that. So just a show of hands again, who is here for biochemistry? Cool. Who is here from Dr. Hashim's class? And then anyone from Dr. Kasuli's? Cool. That makes this easier. Okay, so where to begin? So disclaimer, I'm a student. I am not your teacher. I haven't seen your final. I didn't even technically take biochemistry of Dr. Hashim, so this is just a guess of things I think are important, but I can't guarantee that any of this stuff will or will not appear on your final. So just as like a recap, this is like a best guess. So with that in mind, if there's any errors in the study guide or anything like that, please, take this at your own caution. So I went chronologically on the exam. If I'm correct, your final is 40 multiple choice questions. So since we had 4 exams, you can probably think of it probably 10, 10, 10, right? So each unit. So I'm hoping that the new stuff is pretty fresh in your head, so we could start on the old stuff and kind of work our way there. So if you guys are ready, we can get started. All right, so I wrote up here for Unit 1. I didn't really write a question on this, but don't forget to remember the differences between a primary, secondary, and tertiary, and then I didn't write it, but quaternary structure. Do you guys remember what those differences are? No ideas? Primary, or how about this? What defines a primary structure? If you were looking at like a protein, right? What would it be made up of? Amino acids. Cool. So what about, by the way, can you guys see this? Maybe I should zoom in a little more. Oh, might have zoomed in too much. Is this better? Cool. So what about your secondary protein structure? What would that be made up of? Cool. And what kind of bonding do we see with that kind of stuff? Or how about this? Where do we see covalent bonds? What type of structure? What about disulfide bonds? Where do we see those? You got this. So, disulfide bridges, what are the 2 things between? 2 sulfurs. So we have a sulfur and a sulfur. And what amino acid has sulfur? Sorry, what? Cysteine? Are we in agreement? Cool. So with the 2 cysteines, is that going to be a really strong bond or a really weak bond? Strong, right? So where do you think we really need those strong, strong bonds? What? So firstly, what's the kind of bond that connects two amino acids? Peptide bonds. So if you're connecting amino acids, what kind of structure is that? I think I saw you say it. Primary, right? So peptide bonds. And that's a strong bond, right? What's our strong bonds called? Covalent. Cool. So if we have our strong bonds that are covalent and they're holding the amino acids together, that must mean, what does that say about our primary structure? Yeah. So what about if we have, what kind of structure is it when we have multiple peptides? What? Yeah, polypeptide. What about if we defined it in terms of primary, secondary, tertiary, or quaternary? What about the example in class, insulin? Do you guys remember insulin? What was insulin made up of? We had those two alpha and those two beta chains. So if we have two alpha and two beta, what kind of structure is that going to be? Will it? What did we just say was secondary structure? An alpha helix, yeah. So what if we had two alpha helixes? What does that mean? Tertiary. So two alpha helixes. And those disulfide bonds, where did we see those again? Yeah, between cysteines. So if you could imagine, like, let's say one of my arms is a cysteine and another arm's a cysteine, or has a cysteine on it, and is a polypeptide chain, right? And if they're like this and they're interacting, what kind of structure do you think we'll see there? Yeah, tertiary. So cysteine, cysteine, so that's where we'll see those strong types of bonds. So no matter how complicated biochemistry gets, just going back to the basics and really having that deeper understanding of what kind of bonds we see is going to be really important. So for the first multiple choice question, we have an endergonic reaction with a blank delta H and a blank delta S can be changed to an exogonic reaction by decreasing the temperature. So firstly, what does exergonic mean? Releasing energy. What about endergonic? Needs energy. In terms of sign, what kind of sign would we expect to see? What did you say? Exogonic was what? I'm sorry. Yeah, so because negative is always releasing energy, right? So then endergonic has to be what? Yeah. So what equation, not that we do math in this class, but it's good to understand where the formulas come from, just so we can relate terms. Do you guys remember the equation that relates delta H with delta S? Delta G equals delta H minus T delta S? Cool. So with this equation, When we have negative energy being released, what is that also called? What did you say? I'm sorry. Exothermic. Would it be spontaneous or non-spontaneous? Yeah. Cool. So, I don't know if you'll be able to read my handwriting. By the way, what is this delta H representing here? Yeah, I heard it. Enthalpy. What about the delta S? What is that representing? Okay, I think I heard entropy. And then what about delta G? Yeah. So in order to make this a positive reaction going to a negative reaction, and we know we're decreasing this T value, so this is going down, what do you think we're going to have? Well, if this is a minus sign and we're making this T down and we want this to be negative, then we probably want this S to be positive, right? Because we want to make this as negative as possible. And then this delta H, there's no negative sign in front of it, so what do you think we want to do with that? Make it negative, yeah. So that's exactly what we want to do. So we want to make this negative and this positive to make our number the most negative it can be. So D. Cool. Oops. So another thing that Dr. Hashim really likes is, again, like we were talking about bonds and the interactions. So how does the hydrophobic effect influence the structures of large molecules? Hydrophobic, is that water-loving or water-fearing? Fearing. So anti-water. So if we're water-fearing, nonpolar, is that, or hang on, a better question is water polar or nonpolar? Yeah, water is polar. So if water's polar, our first answer choice, nonpolar molecules are not easily solubilized in water and aggregate. Is that true? Yeah, because they're not going to want to be in the water, so they're going to go away. All right, so polar groups are oriented on the screen, are oriented on the surface, interacting with water. If we have like a lipid bilayer, right? What's on the outside of that bilayer? Water, and it's polar, right? So our polar groups are probably going to be facing polar objects, right? So that looks good. So far, so good. So nonpolar molecules can mask polar characteristics and of the hydrophilic molecules. Can you make something that's hydrophilic look hydrophobic? No, right? So that's not right. So A and B look good. So I think D is a good answer choice. Okay, so the next question is giving us a pKa1, pKa2, and a pKr, so the side group of arginine. And it's giving it to us at 1.8, 9, and 12.8. So at a pH of 7, where would we see the charge on each of the parts? So in case you guys don't have them memorized already, I cannot stress how important it is to have your amino acids memorized. So here's a chart. Oops. So if we look at arginine, and then let's see if I can pull the question back up. Okay. So here's there's lysine, arginine, there we go. So we're given the pKa1, the pKa2, and the pKar. So if we have 1.8 for pKa1, what part of arginine is probably that 1.8? Would it be the carboxyl, the amine group, or the R group? What does it mean if something has a low pKa? What? I think I'm embarrassing you guys. I'm sorry. So with pKa, if the pKa values are low, that typically means it's going to donate the hydrogen straight away, right? So low pKa's donate hydrogens. So with our, if we're donating a hydrogen, that means are we going to become more negative or more positive if we get rid of a hydrogen? I saw someone say it, negative, right? So if we have neutral and we have negative, for our carboxyl group, which we know in general, those are about 2-ish, so 1.8 fits in that. And then we have our amide group right here. So for our amide group, you see how like the carboxyl group here has a negative charge and the amide group here has a positive charge? So the pKa is a lot higher. So species with a positive charge are typically going to be either neutral or positive, which is what we see at the pKa of 9. And then for its side chain, which right here is really high, it's 12.48, 12.5, that's like, that means this is a really acidic amino acid species, right? Because the higher our pKa, the more acidic we are, right? Because we're less likely to give up those hydrogens. So if we have these, oops. The alpha carboxylate, that was the group right here. Its pKa was 1.8. So if we have the pKa of 1.8, what kind of charge should we expect to see? Negative, right? So that gets rid of, oops, that actually gets rid of all of them. Wow. But if we wanted to, the alpha amino group, because we're at pH 7, right? So if this is at pH 9 and this was at 12, that still makes sense, right? Because we're still right here. So that gives us our answer. All right, so number 4. A tripeptide contains 4 ionizable groups with a pKa's of 2. Alpha carboxylic acid, which we said had the negative charge on it, right? Either negative or neutral. So neutral or negative. The side chain aspartate, 3.9, that looks pretty low, right? And if you look here in the book, oops, sorry guys. Yeah, right here, that's pretty low. Looking over here, that's a negative charge. So we can treat this like it's neutral or negative, right? And then 9.9, that's lysine. So let's see, over here. Let's see. Okay, so the group, that's pretty high up there. And we're seeing a positive charge, so we can assume that that's going to be neutral or positive, right? And then the alpha amine group, which we said in the previous question was either neutral or positive. So by taking that, if we wanted to find the PI, what's the goal of the PI? Oh, oops. Sorry, guys. Can you guys hear me better now? Okay. It slowed down my thing. Okay. So if we wanted to find the PI, what is the goal of the PI? Yeah, we want it to be neutral, 0. So let's see. We have one group that can, no, we have 2 groups here that can be negative and 2 groups that can be positive. So we could either have 2 negatives, 1 negative, we could be neutral, we could be positive 1, or we could be positive 2, right? So at what, what's it called? pH are we going to be the most positive? A low pH or a high pH? A low pH, right? Because we're going to be really acidic. So if we wrote the pHs out, so before 2, we're going to be plus 2. Before 3.9, plus 1. Before 9.9, 0. And then before 10.5, negative 1. So then we could add these up, and in my head, because I don't have a calculator, I'm just going to round this to 10 and 4, which 10 plus 4 is 14, right? Divide by 2, it's going to be about 7. And then which of these answers are closest to 7? C, right? Okay. So, again, Dr. Hashim doesn't really have you guys do math, but I just brought this question in just to remind you guys about the Henderson-Hasselbalch. For Dr. Kasuli's class, it's a really, big, important thing to be able to do problems with this. But for your class, just like a generalized understanding is helpful. It's good to know. The Henderson-Hasselbalch, what relationship does that equation give? Do you guys remember? What? Yeah, so it looks like this, right? And hang on. So out of all of these, E says it right here, relates the pH to the solution to the pKa and the concentrations of the S and conjugate base. So there we are for 5. All right, number 6, reaction by which chemical energy has been stored at high energy phosphoanhydride bonds and ATP and is released. How do we, firstly, what does ATP look like? Yeah, so we have our phosphates connected. And then it's connected to a sugar, which you guys can draw. And depending on what it is, if it's GTP, we're going to have a guanidine, right? ATP and adenosine. So the phosphoanhydride bonds, what group is the phosphoanhydride group? Do you guys remember anhydrides from Orgo? It's when you have two things together, right? So if we're cleaving that phosphoanhydride bond, we're cleaving it right here, right? To release all that energy. So how do we typically cleave things in biology? What do we have all around us in the cell that makes cleaving things really easy? We have water, right? So typically, you'll see ATP hydrolysis to release this bond. Okay. While proteins are usually composed of linear chains of amino acid, branched chains of amino acids and internally covalently cross-linked chains can be found in certain proteins. Polypeptide chains are most commonly covalently linked to each other through, this is what we were talking about earlier. Where did we say our polypeptide chains, that higher order structure, what kind of bonds did those have? What? You said disulfide, right? Cool. Yeah. Those were our two sulfurs binding. Where do we typically see hydrogen bonds? This was our secondary structure, right? What kind of bonds do we see between nucleotides in a DNA helix? Do you guys remember? Or what are, let me reword this. What kind of bonds do we see between the bases in our DNA? What? We see the hydrogen still, right? Remember, like, what nucleotide, or what base does A bind to? What? T? And then how many hydrogen bonds are between the two? And then what's our other pair? C and G, and then how many bonds do they have? Yeah. Glycosidic bonds, where do we see those? Yep, sugars. And then peptide bonds, we said we saw those in the primary structure. And then ester bonds are just sort of everywhere, but where do we see that 5' 3' phosphodiester? Yeah, DNA, the backbone. Cool. So Sds Page, this is, again, one of those things where in Dr. Kasulia's class, you'd want to be familiar enough to be able to read the gel on this. But for Dr. Hashim's, I think just having an understanding of how it works is best. So, and should be good. So with Sds Page, what are you measuring? Or better yet, in Sds Page, where does the SDS come from? Do you guys remember? All right, I think I heard someone say it. We have that negative charge, right? And it's sulfur dodecyl sulfate. Don't worry too much about the chemical name. I don't think that will, I can't guarantee anything, but I want to put that on top priority. But the important thing is we dump a negative charge all over this, right? So if we're dumping a negative charge all over our protein, will our protein have a charge of its own, like whatever its charge was beforehand? Not really, right? Because we're like throwing all that negative on it. So if we throw all this negative charge on it, can we measure it by the charge? No, right? So electrophoretic characters, no. Net charge, no. Molecular weight, okay, maybe. We haven't changed the weight. But the charge, no. Molecular weight of the protein, seems pretty good, right? Affinity of protein for acrylamide. Acrylamide's just the gel it goes through. It's like a material. Do you think, like, that doesn't really tell us, you know, anything, right? So, molecular weight. Cool. All right, so another experiment. Hang on, there we go. Salting in a protein can be explained by, so salting in, we have salting in and salting out, right? So with salting in, what do you think our goal is here? So when we're talking about salting in and salting out, think of the in and out as the protein. So in salting in, the goal is to keep the protein in. And in salting out, the goal is to get the protein out. So do proteins like high salt concentrations? No, right? I mean, our body's not super salty, so why would they want to be super salty? So with salting in, what do you think we need to do to salt concentrations to keep our protein this day? We lower them, right? And then with salting out, we increase them to kick the protein out. It'd be like if you put something really smelly in a room or something and you didn't want to be in it, so you'd like run away, right? So let's see. Salt counterions reducing electrostatic interactions between protein molecules. That seems pretty good to me. What do you guys think? I'm going to take that silence as a yes. Okay. So, all right, which of the following amino acids would be the first to elute from a pH 8 of an anion exchange column? So an anion exchange, we have anion and cation exchange. Cation. Oops, there we go. Oops, there we go. Cation. Oops. Hang on. Can you guys still hear me? Yes? No? Did I knock something over? How about now? Can you hear me? Did I knock something over?
Go ahead and turn that one off. I mean, use this one.
Okay.
Is it on?
Yeah, I just turned it off. I feel like the sound just went out. Okay. Is that better? Okay, sorry guys. Okay, so anion and cation exchange. So cations in general, what's the charge of a cation? Yeah, I always think everybody loves cats because they're really cute. So cations are positives. And then anions, that's like anti, right? Like negative. So what's important is to keep those things straight. So with cation and anion, we're talking about the charges, right? That are going through the matrix. So in that case, if a cation exchange, what will the matrix be? The opposite, right? So negative matrix, and then for anion, positive matrix. Cool. So we're giving an anion exchange problem. So if we have our anion exchange column, and we have our matrix in the middle, and we know our matrix has all these positive charges, what's going to stick to it? Yeah, negative charges. So negatives, if they're going to stick to it, are they going to be the first to come out or the last to come out? Last. So what's going to be first, positive or neutral? Positive, right? Because what happens when you have two positive charges? They repel, exactly. So neutral can be the middle. Positives first. Cool. So we're back again with our amino acids. Don't forget to know your the name, primary or one-letter code, three-letter abbreviations, and then it's really good to be familiar with what the different pKa's of the side chains are. So what's the lysine? Is that typically a positive, a negative, or a neutral amino acid? So one of the acronyms I always used was kangaroos radically hop for my acidic amino acids, and then I'd use ED for my negatives. So you can actually see it right here. I'm letting you guys cheat for now. But let's see, lysine is representative of K, so that's a positive. And its pKa, for you guys to know, is about 10 on its side chain. Alanine, where is that? So my analogy for nonpolar amino acids that I used was getting awesome violins isn't likely when my friends pick. That's what I used when I was in the class. And then for all the polar ones, I just kind of remembered it was the last TS, Q, and YC. So if you guys want to come up with a really cool analogy for those, you can. So nonpolar, alanine, that's A, right? So that's going to be a neutral. Glutamic acid, do you guys remember the letter for that? I mean, you can even look at the name. What are acids typically? Negative, right? So they have that pKa, glutamic acid's about 4. And then asparagine, that kind of falls over here. So that's going to be about neutral. And then glycine, nonpolar, neutral. So the first to elute down this column, what did we say was going first? Yeah, positives. So lysine. All right. So which of the following pentapeptides have the highest absorbency at, oops, sorry guys. There we go. Okay, which of the following pentapeptides have the highest absorbency at 280 nanometers? So at 280, do you guys remember the two amino acids that had that high absorbency here? Yeah, tryptophan and tyrosine. And then what had that high absorbency at 260 nanometers? Phenylalanine, right? So when we're looking at the pentapeptides, let's see, we have one tyrosine and then a bunch of histidines. Okay, maybe, right? We have one at the 260 mark. Let's see, this one, we have a bunch of phenylalanines. This would be really good at 260, but maybe not the best at 280, right? So I'm going to keep going. Histidine, methionine, alanine. Not really, we don't have one of our three. Let's see, tryptophan, glutamine, histidine, tyrosine. Okay, that's looking pretty good. What's it called? Sorry about getting these wrong. That's supposed to be tyrosine. There. Oops, sorry guys. There. Cysteine, histidine. I think this was supposed to be a cysteine. Histidine and proline. Not really. So this is going to be our best option, right? Because we have the most tryptophan and tyrosine here. Oops. All right, so we were talking about disulfide bonds earlier. Do you guys remember what we used to cleave those? Yeah, 2-beta-mecaphenol. I don't have this written down, but I would go back during your free time and review those enzyme cleaving molecules. Do you guys remember how we had our trypsin, our chemotrypsin? I think you guys had one or two other ones you had to know, right? So I'd go back. They should be in, I think, chapter 3 or 4 of your textbook. And I would review what cuts after what. And I would also review the danzyl chloride mechanism, which I believe is in chapter 4 of the Leninger textbook. All right. So protein dynamics is the study that examines the movements within a protein. Which type of protein structure determination would be the most useful to study this type of change? So whenever we do anything with X-rays, does that give us motion of any sort? No, right? So both of these, it's like taking a picture, right? And does a picture like give us the movement as something flows? So really the only one that makes sense is B, right? Because this is the only one that would give us a chance to see protein dynamics or any motion whatsoever. All right, so which of the characteristics of a collagen structure listed below contribute to the tensile strength of collagen? So you have collagen and you have keratin. So these are two structures that we spent kind of a bit of time in class going over. And I would go in your book and make an outline of the two differences and how they compare for your own knowledge. But a triple helix where free left-hand helixes twist together in a right-hand sense, is that true? Yeah, right? And then, do we have 3.6 amino acids per turn? Right? Would having a glycine, do you think that would add rigidity to the structure if we had a glycine every 3 amino acid or every 3 residues? It would, right? I mean, glycine, if you look at the structure, it's it has 2 hydrogens, right? So that's going to cause it to bend oddly, right? Because it's functional group smaller. So, okay. So those all look good. So I think D's our answer, right? But again, I would go back in your book and review the keratin structure before your exam. And then 15, a fast and common method for determining protein concentration in a column effluent. Okay, so mass spec, is that a fast method to determine something really quick? No, right? That takes a long time, so that doesn't make sense. Salting out of ammonium sulfate, is that going to be a quick way to determine our protein concentration? No, it's just going to be a way to purify, so that doesn't really make sense. Drying out our protein and weighing it, does that still give us our concentration of protein? No. Measuring light absorption at 280 nanometers, okay. That sounds all right. And then Edmond degradation, what does that do? it gives us sequence, right? So that doesn't really make sense. So that's how we get our answer. So yeah, that's it for Unit 1. Go through Hashim's practice exams again, and I would definitely use those as kind of a reference, and then your previous keyword terms lists to kind of go off with and form your study guide. So Unit 2, we talked a lot about the Michaelis-Menten equation, and I know your exam is only multiple choice questions, so you luckily shouldn't have a full derivation question like I know you guys had on your second exam. But we talked about the steady state assumption, right? What did that mean? Do you guys remember? So we were talking about how we have an enzyme, right? And then we have a substrate. And when we go forward, we form that enzyme-substrate complex, right? So at steady state, remember how Dr. Hashim in class said if you, like, whatever your reactants were, at what state do they equal your products? Do you guys remember that? What terms you can use to describe your reactants and products equaling each other? What? Yeah, so this way is our association, right? Because they're coming together. And then this is our dissociation constant. But when they're equal at that steady state assumption, right? Because we're assuming it's a steady association to a steady deassociation. So you guys remember the other term from when they're equal? Equilibrium. Going back to Chem 2 a long time ago. So when we look at our answers, choice E, ES breakdown occurs at the same rate as ES formation. So it's important to keep in mind what our order for things are. All right, so our next question's talking about our active site. So our active site, what binds at our active site? Hang on. You guys can still hear me, right? Okay, cool. So what binds at our active site, typically, if we have an enzyme and a substrate? Yeah, so for the enzyme's active site, I should state, what binds at the enzyme's active state, or active site? Yeah, so typically if we have our enzyme and it looks like this, our substrate can bind, and we have two models. Do you guys remember the two models for how they can bind? Yeah, induced fit and lock and key. Perfect. And what's the big difference? What does the induced fit have that the lock and key doesn't? Yeah, conformational change. So if this is our substrate and this is our enzyme, and typically the substrate binds, what happens if an inhibitor wanted to bind to that active site? It could stop the reaction, right? Because it's not going to be able to bind there anymore. So if we have an inhibitor binding to the active site, do you think that's going to be an uncompetitive inhibitor? No. Where would an uncompetitive inhibitor bind? Somewhere else, right? Yeah, so it could bind over here, or at any other site on the protein. But let's see, an allosteric effector. I'm not quite sure what that is. I think that might just be there to trick us up. An inactivator. That's a new one too. What about a competitive inhibitor? Do you think it will compete for that active site? Yeah. And then a cofactor, what is that involved in? Yeah, it's like with our APO enzyme. And when we add this, it makes it the hollow enzyme, that activated state, right? And when we have that activated state, the enzyme can be useful. So we see that with our hemoglobin or myoglobin. Okay, so number 18, given the following data of the rate of the reaction with and without inhibitor, so without inhibitor, with inhibitor, what type of inhibition is consistent with these results? So, let's see. On page 209 of the Leninger textbook, and page 374 of the Voyet textbook, there's information on this. So I would definitely check out these pages. But the big thing is our Km and our Vmax. Vmax, what is that? What does that determine? It's our maximum velocity, right? It's when we hit that threshold of how far it's going to go. And then the Km, that's half of our Vmax, right? So when we have these different values, with a competitive, we have that increase of Km, right? And then with our uncompetitive, we see that decrease of both of them, right? And then we have our mix, which can depend on what's it called? The Vmax is down, but then our Km can depend on the situation, right? So if we're looking here, our Km is going from 0.02 to 0.01. Is that increasing or decreasing? Decreasing, right? So Km's decreasing. And our Vmax is going from 150 to 90. That's also decreasing, right? So if we have Vmax and Km both decreasing, to me that's looking like an uncompetitive inhibitor. What do you guys think? So we flip over B, uncompetitive. So again, definitely check out those pages in the book. And then actually skipping ahead really quick. So if you guys skip ahead to question 29, which is given here. So Km. So if you're given a graph and you have to find it, do you guys remember how to find Km? We look at the x-axis, right? And where it's crossing here, we can take the negative 1 over Km. So the picture I got doesn't have the prettiest numbers, and since your class doesn't have a calculator, I'm assuming your numbers would look really nice. But let's pretend if it crossed over here, and let's say over here was 1 over 3, and we had to take the inverse of that, then, or negative 3, then what would that give our Km? 3, right? So that's how we can find that value. And then what about our Vmax? Do you guys remember how to do that? It's the opposite, right? It's our y-axis. So 1 over Vmax. Oops, Vmax. Here. So if this was free, you could take 1 over free, and then you'd get that value. So that allows us to tell if what these values are, and you can calculate those depending on a graph. So if this is our natural, solution before we add an inhibitor, and if we have inhibitor A added here, when they're in parallel, do you guys remember parallel lines, what type of inhibition that represents? It's uncompetitive, right? And then when we have the overlap at the x-axis between the two, do you guys remember what that is? Or not x-axis. Well, yeah, we can do that first. X-axis. So when you have that crossing at that x-axis, that's going to be our mixed, right? Some sort of mixed or non-competitive. So then what does that leave our y-axis as? Competitive, right? Yeah. So that's how you can tell what these are from a graph. And then again, those pages I gave you in the textbook should help with those. All right. So next we have a heme group with myoglobin binding to oxygen and the function of the polypeptide. So when myoglobin binds to oxygen, the polypeptide provides solubility to the heme group, which is very nonpolar. The heme group, what does that have in it? What's like the atom in the middle, or the element? Yeah, iron. So iron, do you think that's going to be a, what's it called? A polar group? Not really. When you think of polar, you can think of more of like your FON, right? Oxygen, nitrogen, fluorine, that kind of stuff. The polypeptide prevents permanent oxidation of iron too. Yeah, remember how we don't necessarily want it to always be oxidized because we're either going to hold oxygen or we're not. The polypeptide helps coordinate the iron. We have this iron group in the middle, so having our chain is going to kind of hold it in place so it can bind to oxygen, right? And then hydrogen bonds, that was something we also saw. So that gives us E. In the slides, Dr. Hashim has some pictures. So I would recommend going back to the PowerPoint where she talks about this and looking at the picture and comparing the different components. So next you have, which of the following increases the affinity of hemoglobin for oxygen? Does decreasing BPG increase that affinity for oxygen? Yeah. So this is going to be found in the T state. And then when you decrease it, that's going to drive it towards that R state, right? So decreasing drives to R state. Okay. Formation of N-terminal carbonates. If you look at that, that's going to be something that has a lot of positive charges on it, so that's going to decrease our pH. How does pH affect our affinity? We want an increase of pH, right? So that's not going to be it. So have a yes and a no. Increasing the hydroxide concentration. Hydroxides, how does that affect pH? Exactly, increases it. So that's good so far. Increase carbon dioxide concentration. So carbon dioxide is sort of kind of like the opposite of oxygen, right? So by increasing that, we're going to decrease that affinity for oxygen, right? And so that's not right. And then an increase in pH. Well, we just talked about how that's good, so that gives us our answers. So we were just talking about the T and the R state. And again, this is where I'd look at those pictures to see what are the things they're comparing. But in general, the really big thing is that iron group, right? And we have that iron group that's moving in the heme plane. So that gives our answer for D. For level of detail, again, I'm not sure how in-depth Dr. Hashim's going to be of our exams. This might be a little too much in-depth, or this might be the level of depth she goes. But I think the best indicator is like your midterms in general. Your midterms have been not as far in depth as you guys could have gone, but also not, the most broad. So what I would do is look at your midterms and maybe the slightly more broad questions, kind of focus on that. But again, that's only a suggestion. Okay. The classical experiment demonstrating that reduced and denatured RNase A could refold into the native form demonstrates that. So RNase A, this is what we were kind of talking about way at the beginning. We were talking, and this was in, I think, chapter 3 of the literature book. But do you guys remember this experiment? Yeah. So the big thing here is we were able to denature and like destroy all those bonds, right? But what was, oh, okay, thanks. Hey everyone in the back, if we could keep it a little quieter for the SI students, that would be really great. Thank you. So RNase A, we were talking about that experiment and we did a lot of denaturing, right? And so we had our peptide strain, and it looked like this. And it had some disulfide bonds and all sorts of stuff. But then we cleaved those and we got rid of our helixes, right? Our alpha helixes and our beta helixes and that kind of stuff. So what did that leave us with? If you get rid of your secondary structure, what are you left with? Yeah. So we were left with primary. And when the experiment ended, were we able to piece back together what we had? Yeah. So what does that tell us? What's like the, what do you need in order to have tertiary structure? Primary structure, right? Because it dictates how things go. So A, primary structure can determine tertiary structure. So there's a picture in your book that really explains this well. I think it's on one slide. So I would refer to that in general. All right. So an ELISA, or an enzyme-linked absorbent assay, or ELISA. Do you guys remember what this was used for? I'm pretty sure you guys had a test question on this. Immuno, what does that make us think? Yeah, antibody. And then enzyme linked, what do you think we're measuring? Proteins. So radioactive substrate, I don't think so. Radioactive standard, I don't really know why we'd have anything radioactive, right? Aromatic amino acids, no, that doesn't really explain the amino part. An antibody that binds to protein of interest. That sounds pretty good, right? But let's look at E, an catalytic antibody. Well, what would that do? How would that give us anything? So it would be D, right? That antibody that binds to the protein of interest. And when that binds, it marks it, right? So then we can see it. All right, so which of the following is false about sickle cell anemia? So there's a glutamic to valine single amino acid substitution. Does that happen? Yeah. There's the hemoglobin S state, which is long and narrow in the hemoglobin. Does that happen? Yeah, that's like the sickling, right? Our blood cells go from happy circular blood cells to unhappy sickled blood cells. And because of the crescent shape that S forms, it gets caught on everything, right? And that's why it's painful. It's heterozygous in those who carry the disease. Is that true? It's homozygous recessive, remember? All right. Carries the disease, this should say carriers of the disease have immunity to malaria. Yeah, so this was, do you guys remember learning about that heterozygous effect? And how you can have an advantage for having one of the alleles to a gene. So that's what that is. And so that looks good. Having higher levels of gamma fetal hemoglobin carriers can reduce pain and symptoms because it dilutes the sickle state. If you have the gamma fetal blood, is that going to be in a sickle shape? No, right? Because it's like the blood you have when you're a baby. So if this is going to be normal, will that dilute your state if you have some normal blood mixed in with your sickled crescent-shaped blood? Yeah. So that's what this is saying. Again, I wouldn't worry too much about this level of detail, but maybe more of knowing it's homozygous recessive and you have like a valine substitution. That's the level I'd be most concerned about. So 25, again, this is one of those things where I would review serine proteases depending on how much time you have due to your exam being on Wednesday. But let's see, you guys had your serine proteases, you had your lysozymes, and then you guys had another pathway you had to know, right? In your unit 2? Do you guys remember what it was? I'm blanking right now, but review your key term list, and whatever's on there, those are the pathways I'd review when you have the option. Again, your level of detail, I would, really the biggest thing I would know is we have the highly conserved proteins, right? That's like the important thing. And the catalytic triad is that aspartic 102, that's right, histidine 57, and then we don't have, this would be a serine, because it's called a serine protease, right? So we have to have a serine somewhere. So, what's it called? And then we use hydrogen bonds. So, again, just kind of where I would focus on things. All right, so which of the following primary structures of an enzyme would the function of that enzyme be most effective by change in pH? So with this question, I realize the way it got cut off is kind of funky, but the important thing here is to note your amino acids and knowing those pKa values, right? Because if you're a neutral amino acid, Like I mentioned, the getting awesome violins isn't likely when my friends pick that abbreviation. Your pH isn't going to affect you as much if you're acidic or you're basic, right? So in this case, let's see. The first one was a glycine, valine, alanine, phenylalanine, and valine. There we go. Do those have any charges? Not really, right? They're all, so we can exit that. The next one, leucine, valine, glutamic acid, glycine, alanine. So we got one there. Alanine, phenylalanine, valine, serine. No major positive or negative charges, though serine is polar. Lysine, we got arginine, we got histidine, and then we have the lysines. So this is so far looking like our, most affected. And then valine, phenylalanine, or alanine, glycine, tryptophan. Is that going to be as affected? No, right? So the one, again, being familiar with your pKa values can really help. Okay, so next up, which of the following about carbonic anhydrase is true? Select also that apply. So earlier we were talking about zinc as a cofactor. Do we see that in carbonic anhydrase? Yeah. What's it called? If present, leads to an increase in pH. Does it affect the pH? What? It doesn't increase our pH. What about this? Accelerates the formation of bicarbonate, which leads to the induction of the T state. How does that sound? Leads to the binding of oxygen in the cell. Would this affect oxygen binding to the cell in a hemoglobin or a myoglobin? No, right? Interacts with free amino residues and water within its active site. Would we see that? Yeah. So in your book, I want to say, I have the Voyet book right now. But you can see it here. Here's your imidazole rings. Here's it interacting with your CO2 and then water. So I'd be familiar with this reaction and the components going on there. And then your imidazole rings, they're part of those histidines interacting with it, right? Okay, so number 28. So when we look at this graph, we have cooperative binding, and that gives us a different curve than if we don't. So what does cooperative binding mean? Yeah, so once one thing binds, that increases its affinity for additional things to bind, right? So that's why with our hemoglobin, when one thing will bind, it will increase its affinity for oxygen, because then it's more likely for two things to bind and so on. So with our affinity and cooperative binding, would we expect to see a hyperbolic or a sigmoidal curve? The sigmoidal, right? So number 2, because we're getting more and more likely to bind as more things bind to it, right? So that tells us that number 2 has to be hemoglobin, right? Because hemoglobin's what interacts with those different oxygens to allow for that cooperative binding, whereas myoglobin has to be number 1, right? Because it doesn't have that interactions anymore. So let's see. That means this is right, right? So KD, we were talking about that dissociation constant. And when does the KD occur? At that 50% mark, right? So if we find the 50%, we can go down for both of them, right, to find our KD. So for myoglobin, that's like, what, about 5 torr? And then for hemoglobin, 30-ish. So let's see, KD of myoglobin, 5 torr. Hemoglobin, 85 torr. No, that takes us way over here. That doesn't make sense. KD for myoglobin, 60 torr. That doesn't make sense either, right? So that gives us our answer. Again, this just shows us how we could have numbers in a question without having mathematical problems. And that's pretty much it for Unit 2. So moving on to Unit 3. You guys had a list of sugars you guys had to memorize, right? Yeah, so I would return back to that key list. And I would make flashcards of whatever sugars you guys need to still have down. It wasn't too many, right? Wasn't it like maybe 6? Yeah. So galactose was one of them, right? Do you guys remember what a D-galactose looks like? It's this one, right? We have the OH over here on the right side. And then we have that, what group is up here? What? All right, so there's either an aldehyde or a ketone, right? So the ketone's between 2 R's, and the aldehyde has a hydrogen on the ends. So when we look at this, that tells us it's an aldehyde, right? So because we know we have an aldehyde here, and then our OH is here, where's our anomeric carbon? Right here, right? So then this can kind of loop around and attack here. And then that would let us draw that cyclic form, right? So let's see. We can start labeling carbon 1, 2, 3, 4, 5, and 6. All right, so we know we have an oxygen. We have that carbon going up here for number 6. Okay, so carbon 1. Do we want to make this an alpha or a beta? What are you guys feeling? Alpha? Okay. So with alpha, how do we know if it's alpha? Is that the same or the opposite direction? Alpha is like their enemies, right? And then beta, their besties. So if you look at the 6th carbon and the 1st carbon, if they're in opposite directions, that's going to be our alpha, right? Whereas if we're beta, we're besties, so we both want to be pointing up. So if we're making this an alpha, we can draw it down like that. So our 2nd carbon, if things are on the left, do they go up or down? They go up, right? So left equals up, right equals down. So let's see, that's on the left, so hydrogen up, OH down. Then OH is here, so we can put that up and this down. And then OH is here. And that's how we do our cyclic form. So again, this test is still multiple choice, but she could potentially give you your Fisher projection or your linear form and ask you to identify this cyclic form. So don't forget how to do those. And I would do that with your key term list of sugars that she gave you from the previous exam. All right, so let's see. In the Watson and Crick model for DNA structure, All right, so Watson and Crick. With the DNA, how, what's it called? What are our base pairs? We said them earlier. Yeah, AT and CG. And then what kind of bonds do we have in between them? Yeah, H bonds. So which ones are our purines? Yeah, I saw someone mouth it. So purines is pure as gold. So that's our adenosine and our guanine, right? And so those are the ones, are they 2 rings or 1 ring? 2, yeah. So again, you guys, I know you had to know your structures for exam 3, and out of all your structures you had to memorize, I'd say knowing your 4 nucleotides in DNA plus uracil are probably like your most important structures to keep in your head. Not only will they be important for your exam on Wednesday, but they also are just really good to know in general for your other classes. So let's see. A purine on one strand always hydrogen bonds with a purine on another strand. Does A hydrogen bond to G? No, that's only if it's like mutated, right? The 5' end of both strands are at the one end of the helix. Is DNA parallel or antiparallel? Yeah. So if it's antiparallel, are those 5' ends going to be on the same side? No. 5 to free, 5 to free. The bases occupy the interior of the helix. Well, I mean, our backbone's made of those phosphodiester bonds, and the bases are in the middle if you, like, draw it out. So, I don't know. That's seeming pretty good to me right now. But let's see what else there is. AT has shares free hydrogens. No, that's 2, right? So I'm thinking C looks pretty good. So another thing she could ask is, she could ask about your AT and your CG content. So with AT, when we have that, if we had a high AT content, how would that affect melting temperature? If we had a high AT content, how would that affect melting temperature compared to having a high GC content? Yeah, it'd be lower, right? Because we have more bonds between here, so it's going to take more to break this. So higher melting point, and then lower melting point. And we want sometimes to have that lower melting point, right? Because if we constantly had all GC, not only would we not have diversity, but then we couldn't zip and unzip our helix together, right? Because we have to replicate our DNA. Okay. Oh, I'm sorry, friends. I just realized I kind of wrote over the next question. What group is attached to the pyrimidine ring in thymine that's not present in uracil? So this is a straight up memorization. Do you know the structure question? So hang on. I don't think I have a picture for you guys right now, but we said our purines had the 2 rings, right? So our pyrimidines have to, oh, can you hear me? All right. So our pyrimidines have to have 1 ring, right? So if you look at the structures, the big difference is that methyl group, correct? Yeah. Okay. So which of the following can be extended by RNA polymerase? So, DNA polymerase. Sorry, got too excited. We don't really do transcription in this class. So, with DNA polymerase, we're always adding in what direction? 5 to 3, right? So if we're adding 5 to free, those new nucleotides are getting added to the free prime end, right? So which of the following can be extended by RNA polymerase? A single strand of DNA ending in a nucleotide with a free prime OH. That's looking pretty good so far, right? I'll put that as a maybe. An RNA single strand, hang on, slow down. RNA, we were talking about DNA polymerase, right? So we can get rid of that. A single-stranded DNA end of a deoxyribonuclease of a 5' phosphate. We just said we were going to add to the free prime, right? An RNA single strand. We just said we were doing DNA. All of the above, no. So that leaves us with A, right? All right. Okay. Inside our protein, or inside our cells, free nucleotides almost always associate with, so we have proteins, we have cholesterol, magnesium cations, fatty acids, and chloride. So do our free nucleotides always associate with proteins? Not really. Where do we find cholesterol? That's usually in like our lipid membranes, right? So we wouldn't really expect to see that floating around with free nucleotides. What about magnesium? What's magnesium's effect in the cell? It's a stabilization mechanism, right? So that sounds pretty good to me so far. What about fatty acids? Yeah, not really, right? Because it's like lipids. Why would a lipid be, or a storage molecule, like a triglycerol, why would that be involved with nucleotides? What about our negative counterions? Again, that doesn't really make sense either, right? The backbone of DNA, what's on it? Phosphates, right? And that backbone, because of those phosphates, you have all those negative oxygens. And if you have negative oxygens, that means the backbone charge is going to be negative. And do you think negative counterions are going to want to interact with a negative backbone? No. And that actually helps support our magnesium idea with that positive charge acting as a stabilizer, right? All right, so which of the following nucleic acids provide the key to decoding genetic information? So decoding genetic information. When we decode genetic information, what's the goal? Why do we have genetic information? What are we trying to make in the long run? Protein, right? Because protein are, for the most part, are functional units. So if we're trying to make protein, which of the nucleic acid provides the key to that? So this is kind of going a little in depth for where the course is, but the answer is tRNA, because what happens is during translation, the tRNA is going to bring those, like kind of looks a bit L-shape, and it's going to bring your amino acids to your mRNA, right? And when it brings those in, it's going to create your new polypeptide structure. So the other reason I brought this up is she could ask you about the three types of RNA, which are listed here, rRNA, mRNA, and tRNA. She could also, I know on one of her previous exams, she asked you about this shape. So I would be familiar with the generalized tRNA shape. Other than that, RNA and DNA, what's like the big differences? Yeah, single-stranded. And what's the other big difference? Yeah, so the ribose versus deoxyribose. Ribose. And then what did we say was the difference with the, what's it called? The bases. We have the U instead. All right, so what's the definition of palindromic? It's like, it's when it repeats back and forth, right? So it's like you could literally like fold it over and it would look like the other side, right? I think race car is an example of this, right? Because like race car, but then you see how like the R's are the same, the A's are the same, the C's are the same. So which of the DNA sequences are considered palindromic? So if we look at this, does G match with T? No, so we can already exit that one out. Does A match with T? Yeah, okay, cool. We have another AT, that's the right pairing. What about G and C? Do those match? Yeah, so this is looking pretty good so far, but we can keep going. Does G match with A? No. Does A match with A? No. A match with C? So that's how we find that. All right, so double-stranded RNA molecules can be cleaved at a specific recognition site by, does RNA polymerase cleave? No, right? So you have RNA polymerase and you have DNA polymerase. And RNA polymerase we're adding during what time? Replication or transcription or translation? Transcription, right? You can think RNA polymerase, we're making RNA. Now DNA ligase, a ligase, what happens when you ligate together? Yeah, so that's a sealer. If we're sealing it, is that cleaving things? No. Okay, cool. Reverse transcriptase, that's when we kind of go backwards, right? And we make RNA go to DNA. Is that cleaving anything? No. Type 2 restriction endonucleases. What happens whenever you see an ACEs? Yeah. So that's looking pretty good. And then polymerase, does that cleave? Yeah. So we see that in DNA replication. All right. So in molecular cloning, transformed organisms must be identified. One common method for accomplishing this involves the inclusion of, what's a way we can get rid of incorrect plasmids and single out the ones that have the gene we want? We could add something that kills the other things, right? So let's say we have a plasmid, right? And let's say I want to insert something that protects the plasmid I want. So a restriction site, that's not really going to help me protect my plasmid or single it out, right? The nuclease gene, that wouldn't, that would just be adding whatever genetic material that would be to destroy it. We have an ACE. A deletion, I don't think deleting anything is going to protect my plasmid, right? What about an antibiotic resistance gene? What if I gave myself resistance and then I put myself in that antibiotic? Do you think that would kill everything else that didn't have the resistance? Yeah, right? Because if you have resistance and nothing else does, it's going to kill everything else. So that looks pretty good. And then an origin of replication, that's where, so plasmids are circular genetic material, right? So they have to have somewhere where they replicate because they are not like our DNA. This is a horrible drawing of DNA where we can start at one end. So that's not really going to protect us though, right? Or single us out. So that's how we can kind of zone out into that answer. Okay, so 39. DNA polymerase from Firmos aquaticus is used in PCR. So with PCR, what is our goal? Yeah. Amplification. So if our goal is to amplify stuff and we want to make a lot of it, we have to run a lot of cycles, right? And when we're running these cycles, what's like the thing that's constantly changing? Yeah, temperature. So if our temperature is changing constantly and we're using a polymerase, which is adding to it, what does that polymerase need? It needs to be stable at those high temperatures, right? So that's what's really unique about the DNA polymerase used in PCR. A fun fact, which I know I've mentioned before at SI, but is that someone actually tried to patent the Firmos aquaticus, which is really funny because it's like trying to patent like seaweed or fish or something. You know, it's patenting a bacteria, a living organism, which it doesn't work like that. So that's kind of like biology drama from the 80s. Thankfully, it didn't happen, or else it would science would be a lot farther behind than where it is today. So Chargaff's rules. So he was the guy who was talking about our different base pairs, right? So the amount of guanine plus cytosine in mammalian genomes varies. Doesn't really sound like a specific rule to me. That just sounds like a random fact. DNA is always double-stranded and RNA is always single-stranded. Most of the time, like 90% of the time, that's true, but is that really talking about our bases? DNA contains two groups, a major and a minor. That's also right, but are we talking about our bases? What about in DNA, the number of adenosine residues is identical to the number of thymine, and the number of guanine residues is identical to the number of cytosine. How does that sound? Yeah. And then DNA contains two strands that run in opposite directions. So again, not incorrect, but not really what Chargaff was talking about. All right, so we already kind of talked about this one earlier. Transfer RNA molecules are involved with. Our tRNA, that was involved with translation, right? Transcription, that's where we make RNA, right? So we're making a bulk of mRNA, but we also make tRNA and rRNA here. But they're not really being involved with that, right? They're just being made there. And then in translation, the big ones are the transfer RNAs, again, being brought in, and then that mRNA with the strands. And then, of course, your rRNA is part of your ribosome, right? And then if we looked at our other options, replication, so a DNA replication, that doesn't really have anything to do with RNA, right? And then reverse transcriptase, that's not really using a tRNA. And then post-translational processing, we've already made our protein at that point, so why would RNA affect our protein, right? Let's see. Which of the following are true of sugars? So in sugars, do the L form predominate? No, right? Which form predominates? The D, right? Where does the L form predominate? Which large biomolecule? Proteins, right? B, the anomeric carbon is the carbon that attacks. Is that the one that's attacking, or is it getting attacked? Yeah, it gets attacked. Which of the following is true sugars? Beta sugars have the anomeric end of the carbon going up in the same direction. That was true, right? That's what we were talking about, our betas being besties. Lysozyme can cut both alpha and beta bonds. So this is kind of referring back a little bit to Unit 2, but lysozyme, that cutted those beta 1,4 bonds, right? And due to enzyme specificities, a lot of enzymes only cut one or the other, right? So if you have something that cuts an alpha 1,4, that probably won't cut a beta 1,4, but vice versa. So that's how we can kind of exit out of that. And then alpha 1,4 bonds are typically seen in branching between polysaccharide chains. So if I have a chain of sugars that go like this, and they link up to another chain of sugars over here, which one of these, what's it called? What kind of bond is between our two sugars typically? Like if we had like an amylose or something. Typically we see alpha 1,4, alpha 1,3 bonds, right? And then our 1,6 bond, that's what was involved with our branching, right? And then which of the following are reducing sugars with only one reducing end per molecule? So this just comes down to purely having these memorized. I'm pretty sure these were all on your list, but again, refer to the list that Dr. Hashim gave you. Sucrose will be the answer to that, because if you look at the molecules, reducing sugars, you need that free anomeric carbon, right? And sucrose has that interaction between the two anomeric carbons where they can no longer interact, right? And then if they can't interact, you don't have any more free anomeric carbons, so you don't have a reducing end. So typically, if you have a chain like this, and let's pretend, for the sake of my diagram, this is, I don't know, glucose. This is just my squiggle saying the chain continues onward. But you would look on this end, right? Like right here at this point to find your anomeric carbon. So definitely be familiar with that. All right. So for 44, so we have a radiogram that was obtained from DNA sequencing, and these give us our dideoxynucleotides. So with a dideoxy, what did those cause? Yeah. 43? Yeah. Oh, oops. I was thinking the question said non-reducing. That is my bad. Yeah, so these are your reducing sugars. See, this is why you need to read the question. All right. Okay, so our dideoxynucleotides, we talked about those, and those were the ones that terminate, right? So if these are our terminators, why do they terminate? Do you guys remember? So if we have ribose, hang on, this is a sad-looking ribose. Or not ribose. So ribose, we have an OH and an OH, right? And then deoxyribose, deoxy means no oxygen. So we have an H at the 2' and an OH at the 3'. But with deoxy, we have 2 deoxys, which indicates the complete lack of oxygen, which means both of our 2' and our 3' on the sugar are both hydrogens. So if our 3' sugar is a hydrogen and not an OH group, can this form that phosphodiester backbone anymore? No, exactly. So since this can't form that phosphodiester backbone, then it can't continue forward. So that's how we know that this is going to be where you see a cleave. So at every mark on this gel, that's where things get cleaved, right? So like this was the chain until a D-deoxy-A hit it. And at that point where an adenosine should have been, it stopped, right? Because it couldn't go farther anymore. So first, you want to write out the strand from lowest to the top, so C, and everything in between. And I'll just do Okay, hopefully that looks right. I did that a little quickly. But that's just lining everything up on here, right? But does this gel give us the complementary strand or the template strands? It gives us the complementary, right? So then all we'd have to do is we'd have to flip this, right? So if we take our C that binds to G on our 5' end, and then our G binds to C, and then our A binds to T, and then we continue onward till the end to get that template strand. So there's 13 more minutes of the review. So for those who come, if you guys want to come up here, I'll give you the answer for the Unit 4 stuff, and then if you guys have any questions, we can go through that material. I can answer any of those. So for 45, I had the answer as A. For 46, I had the answer as A. For 47, I had the answer as A. For 48, I had the answer as B. For 49, I had the answer as D. For 50, I actually didn't work the answer out for this one, but it's asking, hang on. Which of the following peptides listed would you expect to be part of an integral protein? So an integral protein, is that in the membrane or outside on top of the membrane? In the membrane. And in the membrane, is that going to be polar or nonpolar? Nonpolar. So all you have to do is you look at these sequences and you ask yourself, is this a polar sequence? Is this a nonpolar sequence? So again, our nonpolar was getting awesome violins isn't likely when my friends pick. And you can go through, and whichever ones have that high nonpolar content are going to be part of your integral protein. Whereas if you have a high acidic or basic content, or even polar, those are going to be more likely to be on either the edges of your protein or on a different type of protein, right? They wouldn't be in the membrane, because like attracts like. Light attracts light. There we go. Okay, so 51, the answer I had was C. 52, the answer I had was true. 53, the answer I had, it was a select all that apply, and I had A and B. 54, the answer I had was C. 55, the answer I had was B. 56, the answer I had was, A. 57, the answer I had was D. 58, the answer I had was B. B and F. 59, the answer I had was A and D and E. 60, the answer I had was A and C. 61, I had A and C. 62, I had B, C, D, and E. 63, I had the answer as E. Okay, so 64, what defines a primary active transport, to a secondary active transport? So primary active, what do you need to be active? What molecule? Energy, right? So energy, ATP is the big one. Sometimes we'll see GTP or something else. So how could you have a secondary active transporter? Yeah, so the secondary active is dependent on that primary active for its energy, right? And because it's dependent on it for its energy, that's what's going to cause it to still be active transport, because it needs that primary active in order for this to happen. Facilitated diffusion, what is that? What does that require? Some sort of protein, right? So it could be a carrier protein. It could be embedded in the membrane. But that protein is going to help, it's going to go with the current, but it needs to help the ion or whatever you're trying to have cross, right? And then simple diffusion, what's that? Yeah, it naturally crosses on its own, right? It can just simply diffuse across the membrane. All right, so isotonic, hypotonic, and hypertonic. So with our hypo, what do you think when we hear hypo? Low, yeah. What about hyper? Exactly, that increase. So when we're talking about that, you can think of it as we're talking about kind of like, usually it's like a salt or an ion concentration, right? So your iso, these are in balance. So your cell's happy, and the amount of water leaving equals the same amount that's going in, right? But in your hypo, you have a really low salt concentration or ion concentration. So are things going to go into the cell, or are things going to leave? Or I should say, is water going to go into the cell, or is it going to leave? So it's going to go in. And then with your hypertonic, that's when you see the cell like shriveled up because all the water has left because the concentration on the outside is really increased. And so the water leaves the cell trying to dilute that outside concentration, and then it causes the cell to shrivel. Whereas in the hypotonic, your concentration is lower on the is lower on the outside, so the water keeps trying to go in to fix that, and then it causes it to burst, right? So yeah, for those who've come, if you guys have any questions, feel free to come up here. If not, good luck. I hope everything goes really well. I would review again everything from your midterms, your key term lists, everything Dr. Hashim has given you, and thank you so much for coming, guys. I believe that you guys are gonna rock this, and I know most of you guys are returners, so thanks for coming to visit me. Have a good night.